Technology fuels science, but technology is only as good as the people who are trying to apply it for something and figure out big problems and solve the problems with them. What happened usually, traditionally, is that technology is developed in one laboratory and some other people use that technology in another laboratory. But of course, that's not the ideal solution. The ideal solution is when you develop technology in the laboratory where the biological problems are also conceived. It requires special two hemispheres. One hemisphere is filled with technology questions, the other one is filled with scientific problems. Those people who have these two hemispheres are extremely rare. One of them is Karel Svoboda. Karel is a physicist by training. He moved to, after his work and absolutely wonderful work in physics, he worked to Bell Labs. In Bell Labs, he got infected by biological questions. And many of the people who worked at Bell Labs went through this, but only an exceptional few became a real, true neuroscientist. Karel is hard to classify whether he's a neuroscientist. For us, for most of us, he is. But at the same time, he's always doing cutting-edge technology development. He's a group leader at Janelia Farms. He's got a relatively small laboratory. He's a hands-on scientist, and this is his most, I think, with the best strengths, and has got a reputation about that. But he's extremely collaborative. He's collaborating to the rest of the universe, I would say, from Europe to many institutions in the United States, including the Allen Institute. He's a member of the National Academy of Sciences. He's the winner of the Brain Prize. And whenever he's giving a talk, I always consider to quit neuroscience. And I hope this is how you feel after his talk today, but then you have to wait and pause 5 minutes and then go back to the lab. Karel?
Thanks, Yuri, for the inflated introduction. It's really great to be here. Thanks to Yusik and Yuri for inviting me. I've heard about this meeting, and I think this is obviously the development of new recording technologies is one of the key themes that has made neurophysiology advance and will continue to make neurophysiology and actually systems neuroscience in general advance over the next decades. Together, of course, with the data analysis methods, which are probably less of a theme here that many of us are struggling with. So as probably doesn't need to be said here, but the brain produces our perception of the world and drives our actions within it. And so memory, decision-making, and intelligent movements are all produced by sort of core components of behavior. And these behaviors are the result of collective actions of populations of neurons in your brains. And we still know relatively little about how these behaviors are produced by the collective activity of populations of neurons. So we do know that brain functions are localized to specific areas. So there's a sparse subset of brain areas that underlie particular kinds of behaviors. When we dive into the circuits of individual brain areas, we find we're faced with complexity, and this kind of complexity we're now beginning to deal with in the let me see if I figure out this way. In the field, there's tens of, perhaps dozens of cell types in the neocortex, and that are connected in a highly specific manner. Now, these brain regions, in turn, are connected into multi-regional circuits, and it's really these multi-regional circuits that produce behavior. And as you know, information is represented by patterns of action potentials in populations of neurons across these multi-regional circuits. And our work is really focused on computations produced at the level of these multi-regional circuits. So what are the dynamic mechanisms shaping neural representations of information? How these neural representations, these patterns of action potentials, ultimately are related to behavior? Now, we're asking these questions in behaving mice, because behavior allows us to identify, to essentially study core computations, core behaviors. And of course, neurophysiology allows us to get a sense of the neural representation of information, the kinds of computations that might be happening in the brain to support these computations. And of course, neurophysiology has been rapidly developing, in part because of the work of folks in this lab, and really an effort started here at the University of Michigan. And I'll touch on various developments in neurophysiology over the last last decade or so, and also hint at some of the current needs in developing neurophysiology technology. Now, together with modeling of circuits, okay, and manipulations of neural activity, we can try to extract the principles by which these representations are built. And that's something that we're trying to do hard now in the laboratory. And I should mention that we're currently limited in modeling neural circuits and our understanding of the mechanisms underlying these representations by our knowledge about the neuroanatomy, the connectivity of neural circuits. And so the challenge here is that we need to extract principles of neural circuit function with fragmentary, partial information about the structure of neural circuits. All right, today I'll focus on our work on motor planning and short-term memory. And so what is motor planning? Motor planning is really, first of all, about motor control. So many movements, like a baseball pitch, are really too rapid for online corrections. And behavioral experiments going back many decades in humans, non-human primates, we've done these kinds of experiments in mice, have shown that parameters for movements are pre-programmed to make the subsequent skilled movements more rapid and more accurate, okay? So motor planning is an important aspect of motor control, but you can also think about motor planning as having aspects of short-term memory, because the pitcher is instructed by the catcher to make a pitch often long before the pitch, and the instruction and the pitch are related to each other through intrinsic dynamics in the pitcher's head, linking the instruction to the pitch. Both motor planning and the instruction about the pitch are maintained in the head of the pitcher. So you can think of motor planning as having aspects of sort of a prospective short-term memory, linking a past instruction to a future action. And of course, as you know, short-term memory is a fundamental building block of really all cognitive functions. So how do we study motor planning? Well, traditionally, motor planning is studied using something called a delayed response task. This has been studied very heavily in the non-human primate literature. We developed a delayed response task for mice because, of course, that allows us to bring some of the reductionist methods to bear, cell type specific analysis and so on and so forth. So here you have an instruction epoch where an animal makes a decision about a stimulus, perhaps an auditory stimulus or a tactile stimulus. The animal then has to hold that decision during a memory or delay epoch, during which also a response is planned, okay? And then in the movement epoch, the animal signals its decision about the stimulus by making a skilled directional link to one of two targets, okay? So the delay epoch can last on the order of seconds, okay? Much longer than the time constant of individual neurons. So here is a mouse performing this kind of a task under a recording apparatus. Here, this is a tactile version of the task. We won't talk about whisker-based somatosensation. Notice here that during the delay epoch, the animal dutifully holds its tongue before signaling the decision about object location. In this case, this is a tactile version of the task by directional linking. And here is the kind of movements that we're looking at. These are so-called lick rasters, trials arranged in the vertical dimension, time in the horizontal dimension, and each circle here is an individual lick. And mice lick in this rhythmic fashion. This is controlled by a central pattern generator, like the central pattern generator that allows you to walk in a rhythmic fashion. This is in the central pattern generator is in the brainstem, and but the timing and the direction of the licking are controlled by four brain, by cortical structures, as I'll show you. All right, so how do we chase down neural mechanism underlying something like a process, something like underlying something like motor planning? Well, as I mentioned already, there's a lot of evidence from the neuropsychiatry literature and going back 100 years or more that only a sparse subset of brain areas participate in any one behavior. So we have to identify core brain regions that plays roles in motor planning. So we identify the brain area that is critical for motor planning. We refer to this as anterior lateral motor cortex. It's actually one part of a multi-regional circuit that is critical for motor planning. And this ALM, anterior lateral motor cortex, also shows activity that is a neural correlate of motor planning. I'll show you how we did those experiments, and then I'll show you how populations of neurons in ALM and related structures actually shape future movements, are causally related to future movements. You can also say orchestrate future movements. And finally, I'll turn to give you sort of nascent investigations about how these patterns of activity that develop over long time scales are actually generated by the underlying circuit. So let's start with localization of function. We did this in two different complementary ways, and I'll just show you a couple of brief experiments. One is essentially a loss of function screen. We developed methods that allow us to turn off groups of neurons, actually chunks of neurons about 1 millimeter across, with better than 100 millisecond temporal resolution, and we can then in a random access manner using optical methods, this is in the realm of optogenetics, turn off different parts of the neocortex during behavior, during different phases of behavior, during decision making, during motor planning, during movement execution, then ask which parts of the brain are critical for motor planning. Turns out that only one brain region lights up. When silencing this brain region, we get an ipsiversive bias in future movements, and this is this anterior lateral motor cortex. Silencing any other brain region had no effect on behavior. This is sort of a loss of function screen for brain areas that are critical for motor planning. Now, when we then used silicon probes, thank you, Ken, to implant them into ALM, we found neural correlates of motor planning. About 60% of neurons show activity that relates, that is task-related, and that shows dynamics that is consistent with the role in motor planning. Let me show you one example neuron here. There's a neuron, and the trials are arrayed by trial type. These are right movement trials, left movement trials, right movement trials in blue, left movement trials in red for the rest of the talk. Here is when the decision is made during the instruction epoch. You can see that this neuron already becomes selective, okay, for right movements. And then after the stimulus is withdrawn, it remains selective throughout the delay epoch until the selectivity and activity collapses in the peri-movement period. Okay, so this neuron, essentially this activity, this memory-related activity, bridges the time period from the instruction to the movement. Now, you might say, is this really related to the future movement? To address this, you have to look at the error trials where the instruction was to move left, but the animal moves right, and still we can predict on a trial-by-trial basis what the animal will do, where the animal will move, rather than what the instruction was. So the activity pattern really predicts the direction of movement long before the movement, and this is why we refer to this as a neural correlate of motor planning. So we record these neurons in ALM. So the next question, of course, is this preparatory activity, is also referred to as preparatory activity, or the neural correlate of motor planning, is this really localized to the area that we have identified by inactivation, or is preparatory activity more distributed across frontal cortex? So here, this might be a preparatory activity here shown by these magenta triangles, more distributed across the motor cortex, or it might just be limited to the anterolateral motor cortex. So this kind of fine-scale mapping is actually better done with imaging experiments that still provide single neuron resolution, perhaps with slower temporal dynamics, but that gets us dense sampling of neurons in a neighborhood and allows us to identify boundaries between functional areas. So this has been become possible over the last decade or so with the development of new fluorescent indicators of neural function. In particular, GCAM, many of you have heard, is a green fluorescent protein-based calcium indicator. It consists of a modified green fluorescent protein, shown right here. This is the beta barrel of the fluorescent protein, with a calcium binding moiety that when calcium comes in, neural activity associated with increases in calcium in the cytoplasm. When calcium comes in, calcium binds to the calmodulin right here on the C-terminus of the beta barrel that then wraps around the calcium binding peptide, plugs a hole in the side of this fluorophore and then produces a bright green fluorescent protein from a dim fluorescent protein. Now, these fluorescent indicators have been around for a long time. The initial kind of scaffolds were based on green fluorescent protein and were developed right after the cloning of a green fluorescent protein more than 20 years ago now. But they were for about a decade or so, they were much too dim and not sensitive enough to be used for in vivo imaging experiments. So this really called for an engineering effort to improve these fluorescent proteins by factors of 2 year after year to ultimately move them into challenging in vivo situations. I'm bringing this up because it's a kind of challenge now that is faced by extracellular neurophysiology also. There's a great value to actually these factors of 2 improvements year after year, because very soon you have a factor of 8 and a completely different set of experiments is possible. And this is really what happened here with GCAM. And this is really now the result of a project, the GENIE project at Janelia. There's a bunch of lab heads involved and technical staff. It's essentially like a biotech company running at Janelia with the business model is to lose $1.5 million a year or so. And all the output of the biotech company are reagents that are widely available, freely available at Atgene for plasmids, JAX for mice, UPenn Vector Core for viruses, and so on and so forth. And what we do is take scaffolds for indicators that report neural function. This is GCAM, and we also work now on voltage sensors, and then put them through this loop where we stare at these sensors, do structure-guided mutagenesis, tweak them in various ways, often semi-randomly, then transfect these sensors into neurons, do 96-well screens, test them in imaging assays in neurons, and go through the cycle over and over again. And after some time, we then filter out the best sensors and test them in other systems like mice and flies in vivo for better function for reporting neural activity. So I'll just show you one data set. This is the most recent GCAMs. These are GCAM 6s. These are the most widely used protein sensors in the laboratory. And now the data that underlies the development of GCAM 7s. Okay, this is about 1,000 detailed biophysical characterizations. And these sensors have recently been made available, and we're still writing up the paper. So using these kinds of sensors now, we can read out activity at the level of individual neurons, now using imaging and fluorescence. This is how this looks. This is actually still done with GCAM 6. The GCAM 7s are so new, I haven't had time to make a movie. Here, Lucille cell-attached recording, extracellular recording showing the spikes in red, and the fluorescent strangens here corresponding to this recorded neuron in yellow. And at least in favorable conditions, when you sort of zoom into one neuron, you can make a good correspondence between action potentials and fluorescence strangens. So by looking at the fluorescence dynamics in yellow, you can learn about a lot about the spikes. And this is, of course, useful because now we can not look only at one cell, but at large populations of neurons using imaging approaches. In fact, the development of GCAMs has enabled new types of microscopes. I think Mike will talk about a different approach to microscopy. Here, we developed a bezel scale microscope that allows us to do this kind of experiment that we set out to do, to map neural activity at the level of individual neurons over multiple brain regions. So here we had to break essentially the constraint imposed by traditional microscopes that either allow you to look at with high resolution at small field of view or low resolutions at large field of view. This is a microscope, the so-called mesoscale microscopes, that allows you to image with reasonably high resolution at large fields of view encompassing multiple brain regions. And I should mention this was really spearheaded by Nick Sofroniev, a graduate student in a lab, and Dan Flickinger, an engineer who I've collaborated with for many years. So this is how this looks. The microscope is here hanging above here a floating treadmill that a mice might sit on. Here's the objective of the microscope, and the key specs are that this provides a field of view of 5 millimeters, very high excitation numerical aperture, so the excitation volume, 2-photon excitation microscopy, is smaller than a neuron in typical neuron in the brain, and also high collection efficiency so that we can detect the signals efficiently. So the two key inventions in this microscope are a beam path that allows us to move a laser over a cylinder that's 5 millimeter in diameter, 1 millimeter thick, essentially instantaneously anywhere in that cylinder. And then also a detection path for fluorescence that allows us to collect that fluorescence efficiently. And also perhaps germane to this kind of a workshop, this microscope worked very well when we constructed it, and there was immediately the question, this is a complicated piece of equipment, how do we get it into the laboratories of other folks outside of our lab and outside of Janelia? So we came up with a scheme that actually worked rather well in hindsight, and that was to essentially organize a workshop. So we recruited some sort of cutting-edge imaging lab and asked them if they were interested in cloning this microscope. About 14 laboratories agreed, and they came for 1-week workshop to Janelia, during which we actually built one of these microscopes, and they at the same time ordered all the parts for the microscope and learned about how to use the software, and essentially got instruction on how to construct this thing in their own laboratories from A to Z. And this is a picture from the workshop. And the enthusiasm in turn generated by the workshop in turn got microscope vendors interested, one of whom who licensed the mesoscope. So here there are now 2 ways to get this hardware. One of them is to just take it from the website, build it yourself based on instructions that we developed during the course, or buy a turnkey system from Thorlabs. Now what can we do with this? Now going back to mapping motor planning related activity, this is now, let's finish this a detour on imaging technology. What can we do in mapping motor planning related activity? Now we can look at activity, say, across 4 by 4 millimeters in the cortex. Each one of these, this always goes back to by too fast. These are flashing circles that correspond to individual neurons. Here are the neurons segmented. Here are fluorescence dynamics that where each line here corresponds to the activity of individual neurons. So this now provides a lot of data, spatially resolved, that can provide information about the coding of information across the cortical surface. And by combining this kind of data with behavioral information, we can then ask questions about what kind of features are coded in different parts of the brain. And now here focusing on the motor cortex. This is time within a trial. Negative is before the movement onset. Remember, there's an instruction epoch, a delay epoch, and a movement epoch. 0 is the onset of movement, and what you can see is that stimulus selective activity related to the distance to the object explored with whiskers, comes in through the back of the brain, through the primary motor cortex, and then movement-related activity appears first and is really limited at the beginning to the anterolateral motor cortex, in particular to the deep layers of the anterolateral motor cortex. So there's an excellent correspondence then between the ALM defined by inactivation and by mapping activity. Activity that predicts future movements is really localized, at least in the beginning, in the anterolateral motor cortex. And there are many other experiments that lead us to the conclusion that ALM is really a hub for motor planning, at least for these tasks that involve orofacial movements. All right, let me now turn to how the brain uses actually neural populations for behavior rather than individual neurons. So everything so far was really focused on mapping the dynamics of individual neurons. Of course, the brain really relies on neural populations. How do we make sense of neural populations? And what I'd like to show you is multiple types of experiments of various types that convince us that really the dynamics of neural populations during motor planning orchestrates future movements. So why is this a challenge? Well, because the population dynamics is complicated, right? So here's again the neuron that I've shown you earlier that becomes selective early, maintains its selectivity during the delay epoch, and then collapses its selectivity during peri-movement period. This is just one of many different kinds of response types in the same chunk of tissue. Even though all of these neurons in some sense predict movements, okay, it's often seconds before the movement. For example, different neurons have different selectivities. The rightward selective neurons and leftward selective neurons intermingle on both sides of the brain. There are neurons that are selective early, only during decision making. Other neurons retain selectivity during this memory epoch, okay, or become mostly selective during the memory epoch. Yet other neurons seem to be selective mainly during peri-movement period, consistent perhaps with driving the movement. So how do you make sense of this at the level of populations? And let me now focus on preparatory activity during the delay epoch, okay? And during the delay epoch, it turns out that the activity of most neurons is relatively straightforward. It either ramps up or ramps down up to a movement. And we can make sense of the population dynamics using relatively simple tricks from dimensionality reduction methods. And these are essentially linear algebra tricks that then allow us to interpret how the population dynamics relates to behavior. So what are dimensionality reduction methods? Well, we're typically recording with the silicon probes that we've used here from about 30 neurons simultaneously in the ALM. We now, using neuropixels probes, often record from 60 or 80 neurons simultaneously. But 30 neurons actually is sufficient to estimate many aspects of the dynamics of ALM during this, at least a simple kind of task. So, but the activity is really, you can think of as dynamics in a 30-dimensional space where each axis in this 30-dimensional space corresponds to the activity of one neuron, and then the preparatory activity corresponds to trajectory in this 30-dimensional space. And there's on average, a different trajectory for anticipating different movement directions. So we can then define a coding direction. This is just one way of doing dimensionality reduction that we find especially interesting for explaining behavior. We define a coding direction as the direction that best separates the trajectories across trial types. And it turns out this direction in activity space is stable across time during the delay epoch, and then project activity along the coding direction. And for the aficionados, this takes care of about 30–40% of neural variability. So it explains a lot of the variability, and it really accounts for most of the trial type selectivity in the dynamics. And I'll just focus on this projection for the rest of the talk. There are other projections that are also important that inform us about the timing of movement and so on and so forth. I'll focus on this projection. So naturally, because I projected on the most discriminating dimension, the trajectories projected into this dimension play a part in a time-dependent manner, here in a trial-type dependent manner up to movement onset. Okay, so can we, and it turns out that dynamics in this coding dimension explains many aspects of behavior. So for example, of course, the brain makes use of single trial information. What I just shown you is the average trajectory averaged across trials. But on individual trials, the trajectories vary in a trial-by-trial manner. On the right are some raw data. You see some sampling noise here. This is fast stuff, but there's some systematic differences across individual trials. And at the time of movement onset, the trajectories overlap. Okay, so if ALM preparatory activity instructs future movements, we expect a relationship between the single trial dynamics during the delay epoch and various aspects of movements. And I'll just show you one example related to movement direction. So we expect, of course, that if the trajectory is close to the average right trajectory, that for right movement trials, that right movements are more likely. If, on the other hand, the trajectories spill into left movement trajectory, perhaps left movements seep in with a reduction in right movement probability, and so on and so forth. And conversely, for left movement trials, and this is exactly what we find in the data. So for trajectories that are more similar to left movement trajectories, left movements are more likely for right movement trials, and conversely for right movement trials. Similarly, for the timing of movements, the timing is fastest for trajectories that are close to the average trajectories, and lowest for these more ambiguous trajectories between the two average trajectories. This suggests really that the dynamics of ALM really instructs future movements. Now, of course, it's still indirect. It could be that ALM, this brain area that we've identified, even though identified with loss of function methods, that has its own set of interpretational issues. Perhaps ALM just listens to another brain area that actually controls movements, and the fluctuations are inherited from that other brain area. So we thought is we now produce fluctuations in ALM and see how that changes, to what extent that produces changes in behavior, and these changes in behavior are related to changes in ALM dynamics. I've done this in a variety of ways. I'll just show you two kinds of experiments that kind of straddle the kinds of perturbation experiments one can do. The first is sort of a cudgel, a crude method. Here and I like this experiment a lot, it's quite incisive, I think. You bilaterally inactivate ALM transiently, and bilateral inactivation is required to actually kill all memory about trial type in subsequent dynamics. After transient inactivation, the trajectories just meander randomly in this activity space along the coding dimension. And activity now, again, has no memory about trial type. The animal moves one way or the other, 50-50, independent of what the trial type is. But on a trial-by-trial manner, based on the dynamics of ALM activity, you can still predict what the animal will do depending on the exact shape of the trajectory. So I think this indicates that ALM activity trajectories during the delay epoch direct future movements. Now, as I mentioned, this is the kind of experiment that we did. In this experiment, it would also be informative, perhaps, if we could just tickle trajectories a little bit in a more targeted manner. Here, of course, we inactivated neurons non-discriminately. We relied on the intrinsic circuitry of the system to produce trajectories that sort of made sense, were interpretable, that we could rely to behavior. Can we now tickle these trajectories a little bit and see if we, in a predictable way, push them one way or another, whether or not we see expected behavioral effects? So what we do here is this is now back in the realm of imaging. We map the receptive fields or the dynamics of individual neurons, rightward selective neurons, leftward selective neurons. These neurons also have dynamics. Some are early selective, some are late selective. These neurons also now express channel drops, and we can now use targeted two-photon excitation to stimulate individual neurons based on their selectivity in control trials, and either stimulate neurons that are leftward selective or rightward selective or any combination thereof. And it turns out just by stimulating on the order of 10 neurons or so, often we just stimulate 9 neurons, we activate dozens of other neurons on the order of a couple hundred neurons throughout ALM. And then when we compare the change in selectivity produced by photostimulation now of individual neurons, we find that if we change the selectivity to rightward movements, during the delay epoch, we make rightward movements more likely, and vice versa. These now very tiny behavioral effects consistent with these very tiny perturbations that we produce at the level of the entire network. So by analyzing the effects of different kinds of perturbations on both neural dynamics and behavior, I think we can conclude that ALM is part of a multi-regional circuits that is cause-related to future movements, so which that produces that has dynamics that is causally related to future movements. All right, so let me so we find preparatory activity on in the anterolateral motor cortex that is that directs future movements. And in work that I don't have time to show you, we also found that ALM is organized in two modules on both sides of the brain. And both sides of the brain show preparatory activity for both movement direction with similar dynamics. And these modules on each side are connected by the callosal connection, by the corpus callosum. And I call these modules because each module on the left and on the right can maintain preparatory activity independently of the other modules. So if you perturb preparatory activity only on one side, the intact side maintains preparatory activity independent of through the perturbation, and that actually helps the perturbed side recover perturbations. Now the question that I'd like to address next is how is this preparatory activity, this memory-related activity maintained? So here in this experiment, we varied the delay epoch, and you can see how here neuron that produces persistent activity up to several seconds. There's a rightward selective neurons in one of these modules. How is this activity maintained? This is a non-trivial problem because, as you know, neurons have time constants of milliseconds. If you inject information into isolated neurons, that information decays over time scales of milliseconds. How is this persistent activity maintained? So one way, so this is just what I mentioned, is if you inject different stimuli, for example, sorry for those of you who can't see this down here, when you inject information into isolated neurons, the information decays over time scales related to the time constant of the neuron. This might be, for example, information provided during the sample epoch to the system. Neurons are essentially memoryless components. So how do you produce these persistent states? This has to be an emergent property of neural networks. So one way this has been modeled for decades, going back to the 70s, is through recurrent feedback. And we think that actually excitatory feedback is critical in this system, that neurons are connected in a like-to-like manner, and that you can convince yourself that that can easily prolong the time constant of the system. So here, if a neuron now gets a little bit of input here, and then that input decays, it has excited downstream neuron that now provides some back excitation which prolongs the time constant of the neuron. And using this kind of principle of positive feedback, you can build different models, different kinds of models that work by different principles that can maintain persistent memory-related activity. And many models, dozens of papers here, they really fall into two classes. One is an integrator, which converts a graded transient input into graded persistent input, and this has been most famously modeled for the system that maintains eye position, but also working memory, and discrete attractors, which converts a graded input into discrete outputs. So in our case, this would be different movement direction. And it turns out that by looking at the dynamics of neural populations during the delay epoch, and by also looking at the responses of the system to optical stimulation, we think we can reject this model. And I think among the models, this is the last one standing. So let me give you one key prediction of the discrete attractor model that our data are consistent with, and that is multi-stability. And I think this is the most stringent test of the discrete attractor model, and multiple other arguments that I won't bore you with. This is really the most interesting experiment. So during the delay epoch, these networks should show multi-stability or bistability here for two movement directions. after perturbation, the discrete attractor network, if pushed close to the separatrix, so discrete attractor networks, you can think they converge to wells in phase space, okay, with big energy bumps between wells, and that's shown here in this cartoon. If we push the system a little bit, not beyond the separatrix, the line separating the two wells, the system should decay to the correct endpoint with correct behavior. In contrast, of course, when we push the system beyond the separatrix, it should relax to the other fixed point with now an error in behavior for that trial type. Okay, so can we do this kind of an experiment? Again, here now is preparatory activity, right movement trials, left movement trials, and we transiently, and this is the work of Hidehiko Inagaki, there were a lot of subtleties here. He came up with an optogenetic perturbation, I can't go through the details, that collapses the selectivity. Essentially, you see the red and blue trajectories lie on top of each other, having relatively little effect on mean activity, and things seem to essentially normally recover afterwards with some errors that are produced by the photostimulation. So there's complete collapse of selectivity along the coding direction. And what we expect from models of the integrator is that the integrator should then proceed with essentially the same slope after the perturbation. In contrast, the discrete attractor should snap back to the trajectory it would have had in the absence of a perturbation, at least if the correct endpoint is recovered. And this is in fact what we see in the data. So on correct trials, the trajectory snaps back to the correct trajectories. In contrast, on error trials, the trajectories are swapped. Now the red left trajectory looks like the right trajectory, and vice versa. In either case, the trajectories snap back to the trajectories seen in the unperturbed case. So this bistability is really a signature of the discrete attractor model, and we think that really discrete attractor dynamics underlies preparatory activity in ALM with each one fixed point for each trained movement. All right, so I have another 5 minutes or so, and so what I'll do, yeah, so I just wanted to quickly show you a working model of the system right now. So this is really, this model contains essentially all of the information, and it sort of explains most of the data that we have. We have two attractor networks on each side. Neurons with similar selectivity are connected to each other. This is also based on functional anatomy analysis. There's non-selective inhibition in the system, and one module on each side. And these modules are coupled using weak connections that, again, connect like-to-like neurons across the hemispheres. And this produces robustness of the system. If one side is perturbed, it's rescued by intact preparatory activity in the other side. When an animal is preparing for a movement, for making a decision, we think there's a non-selective input coming into the system. We think probably from the brainstem through the thalamus, that is related to essentially an urgency signal, and we've seen that kind of dynamics in the brainstem. We're working on this now. We have several lines of evidence that there's a non-selective input coming in. This non-selective input has a different slope for different timed movements, more shallow for slower movements, steeper for faster movement. And it's this non-selective input that destabilizes the ALM network, producing two attractors. And it's the selective input related to object location or tone, related to decision making, that biases the system in one or the other attractor. Okay? And so some of these are work in progress, but everything we see is sort of consistent with this kind of a model. And this kind of a model also recapitulates many aspects of the dynamics of the data and also its behavior to various kinds of perturbations in photostimulation. So let me just summarize this part of the talk then, and then turn to some ongoing work and just some final remarks about where electrophysiology might have to go. So we've identified Keynote, the anterolateral motor cortex for motor preparations. What I didn't have time to show you is that we know that ALM acts in concert with the cortico-thalamocortical loop. It's really a part of a cortico-thalamocortical loop that maintains this preparatory activity. And during motor planning, preparatory activity converges really to an initial condition in activity space that allows rapid and accurate movements. And I think that's really the role for motor planning. And reaching the appropriate initial condition is necessary to produce that fast and accurate movement. And the initial condition in activity space, or the final point that motor planning proceeds to during the delay epoch, corresponds to the attractors, to fixed points of discrete attractors. And we think that preparatory activity is implemented in modular and redundant circuits. Now, when you look at models of short-term memory, motor preparation, working memory, folks always think about cortical circuits, intracortical circuits, because we know that cortical neurons are connected to each other. But as I already mentioned to you, we think that multi-regional circuits are involved. And we've been interested in exploring the circuits of the cell types of the ALM. I won't go through great details here. Suffice it to say that there are many, and these cell types are complex. So these are single neuron reconstructions here in the anterolateral motor cortex on one side of the anterolateral motor cortex superposed. And you can see that these circuits are not localized, right? Although there's a lot of local axons, there are a lot of projection across the cortex and the cortical nuclei. So many of these axons are actually striatal. And then, as I already mentioned, there's a strong callosal projection. Actually, the strongest input to one side of ALM comes from the other side of ALM. But these neurons also project to the thalamus. And this is a critical part of the circuit that produces motor preparation. And also, because it's a motor cortex, there are also neurons that project to motor centers, in particular to the medulla, that initiate movements and that control directional licking and jaw movements and so on and so forth. We're really building an atlas of ALM cell types, starting with morphology and also combining morphological reconstruction with the molecular characterization to really get a sense of an inventory of the cell types, and ultimately, of course, to link them into defined circuits, cell type specific circuits. And I think this matters. And I'll just give you one example of some ongoing work. This is work by Mike Economo in the lab. He found that the neurons that project out of the cortex to motor-related centers fall into two classes. These are both so-called pyramidal tract neurons in green. These are, again, single neuron reconstructions. In green are neurons that project to brainstem centers, but they avoid the premotor nuclei related to orofacial movements, but they project to the thalamus right here. And then in red are neurons that project to the medulla, to these premotor centers that we think initiate licking and directional movements and so on and so forth, but these avoid the thalamus. So thalamus projecting pyramidal tract neurons and medulla projecting neurons. And just one data slide here. This is important also in the context of movement initiation and movement preparation, because if we now do cell type specific recording, and I should emphasize that these are really quite heroic studies, as Mike Fee well knows. These are essentially antidromically identified neurons that are recorded in ALM and then stimulated either by their targets in the thalamus or in the medulla to identify neurons that project to the thalamus or to the medulla. And this is worth doing because these different neuronal populations show very different dynamics. So here, this is the movement-related selectivity for thalamus projecting neuron. These neurons become selective early during the instruction epoch and maintain selectivity up to the onset of the movement. In contrast, these medulla projecting neurons show some selectivity, much less selectivity, and selectivity decays. And it's these neurons really that are critical for maintaining the preparatory activity through a cortico-thalamocortical loop. So what this reveals really is that there is specialization at the level of cortical projection neuron types. They're neurons that are perhaps specialized to maintain through feedback from the thalamus preparatory activity, okay? And these neurons also receive input back from the thalamus, so forming this nice loop. And then these medulla projecting neurons that we think are critical for movement initiation. Now, importantly, I want to, one reason why this is important for this community is that this is really only scratching the tip of the iceberg. Here is a hierarchical clustering of now the molecular cell types of cortex. This is ALM and visual cortex combined. This is the work of the Allen Brain Institute that we've collaborated with. So in black are neurons that you find here in ALM, in purple that you find in the visual cortex. Is an extremely interesting paper. If you're interested in this kind of stuff, I encourage you to read it. Suffice it to say there are probably 40 GABAergic neurons, at least defined transcriptomically in the cortex. We don't know yet how many actual cell types that corresponds to. Even the definition of cell type is a little fluid. The way I think of it, it has to have a transcriptomic signature and some other kind of parameters that link to it, anatomy, connectivity, or perhaps neurodynamics. And there are probably on the order of a dozen projection neuron types, both for ALM here in black and visual cortex in purple. Note that all the GABAergic neurons are actually shared between the visual cortex and ALM cortical areas that are on different sides of the brain, different ends of the brain, whereas the projection neuron types are distinct, transcriptomically between ALM and V1. And I think that electrophysiology has to figure out how to deal with this complexity. Right now, we have only very poor methods to link cell type to electrophysiology. And I think for electrophysiology to move forward, we ultimately have to figure out how to recognize different cell types in a more efficient manner than we know now to put the recorded neurons ultimately into some kind of framework of neural circuits. And that's really ultimately, I think, what it will be about in systems neuroscience. And I'll be happy to discuss some ideas about that with you during this conference that I look forward to very much. Okay, let me just highlight the folks who've done the experiments that I mentioned, especially the recent stuff was done by Hiko Inagaki, spectacular postdoc who's worked on principles underlying persistent activity, who came up with these key experiments that allowed us to test different attractor models. Mike Economo, who did cell type specific analysis, also together with Sarah Vishwanathan, Sandro Romani, Lorenzo Fontolan, who were very important to look over our shoulders and help us with modeling. The single neuron reconstructions were done in collaboration with the Janelia Mouse Light Project and the molecular work with the Allen Brain Institute. Thanks for your attention.